SF Hack Night. NeurotechX is an international community that facilitates the advancement of neurotechnology. Our pillars are community, education, and innovation. And we have chapters worldwide, local meetup events, and a thriving online Slack community of, I think, over 4,700. And yeah, if you don't have a chapter in your area, please get in touch. If you're not already on Slack, also check a link in the Meetup or let me know, and we'll get you an invite. And check out our online educational platform and newsletter. Okay. And check out our job boards and other services too. So welcome everybody. This is our weekly meetup, and this is the usual time in the evening. Just ask anybody who's joining for the first time if you don't mind introducing yourself and telling us a little bit about what brings you here. There is no requirement to do that. So
I'm sorry, it took me a minute to unmute.
Oh, sure.
My name's Bill Horn.
Sure.
I work on the UC Davis campus as a nutritionist. In our lab, we have a nutrition and stress researcher, Kevin Leggero, and he's gotten me very interested in using some neurohacking techniques. I've got a little bit of experience with thought technology. I just bought a bit of Lino from Portugal, and very much a newbie, but I just want to play around with some stuff and see what I can find out.
That's awesome. Thanks so much for sharing. And yeah, if you've got a hardware device, we would always love to see it, especially if it's one that maybe we're not familiar with. One of our many goals is to update our online resource to give people. I mean, probably the most frequently asked question is, what hardware should I get?
Yeah.
And there's a lot of devices out there. Certainly, one of the things that we're really interested in doing is giving people access, because it's EEGs really, and NIRS and some other technologies are very accessible. These aren't incredibly difficult systems to build, but they are difficult to use and interpret. So that's super awesome. I just caught a really interesting webinar with John Cryan from I think it's University. It's a university in Ireland. I forget whether it's Dublin or Cork, but talking a lot about gut-brain axis work, and anyway, I've got some. I actually was going to bring this up later, just because some of the papers that they had here was the neuroactive potential of the human gut microbiota, and in quality of life depression, and this particular bacterial strain as a translational psychobiotic, which it sounds, and so it's modulation of stress, electrophysiology. Actually, I'm going to drop this link in because it's super relevant for what Bill just talked about. And what I liked about this was that they were actually doing EEG in VR as part of the this study, which hopefully they'll tell me where this is. Oh yeah, University College Cork. But and let me just give you a link, because you can also catch this same talk, as this was hosted by iScience of Cell Press, and yeah, I mean a terrific researcher, obviously, who's somewhat focused on mouse models, but like everybody interested in translational work as well. But the mouse work that he covered in this talk was excellent, and yeah, so super interested to cover how we can measure stress and its physiological responses.
Yeah, I'm very interested in measuring, vagal responses, and I've had people who told me it's like trying to get water out of a fire hose. There's a lot of signals going through that nerve.
Yeah. So one of the I mean, we're I've got the I've got speaker. I mean, you'll see the Meetup that I've got Meetup events for all of August already posted. And on the 20th, I think we'll have peripheral nerve stimulation researcher coming, grad student.
Good.
Is that some background noise though? Somebody can mute.
It seems like it's coming out of Ryan's, but it also says that he's muted on the microphone.
Yeah.
Okay, I will try and send him a message. It does that, doesn't it? But it says.
I think we can mute him for now if you want.
Okay, let's see if we can do that without muting. I don't know. Can anybody hear me?
Yeah, I can hear you.
Okay, good.
Yeah.
Okay, well, thank you. So, yeah, very, very interesting talk. So, somewhat relates. I mean, there's, yeah, we would love to get more vagus nerve stimulator people to come talk too. I think she's working. Well, I think she's working on vagus nerve, but we'll see August 20th. The microbiota stuff is very Bastian of the director at Open Humans has talked about the difficulty of really capturing your gut microbiome, certainly with currently available technology, which is too bad. I'd still love somebody to come on and say what happened with Ubiome.
Yeah, is this like related to, since we were kind of talking about how food makes us feel? You were just thinking of this stuff, and so more practically, they're looking at. It says the gut-brain axis across the lifespan. Can you elaborate on what the gut-brain axis is?
Well, sure. So one, you have a lot of neurons associated with your gut, and they're referring to this gut-brain connection, and it and again, like what you'll see if you catch this talk is the amount of mouse work that's going on in terms of controlling, raising. I mean, if anybody's familiar with Ed Yong's book too. You contain multitudes, or I contain multitudes. I think it's I. But Ed Yong's book, if anybody can find that link, is also a great intro in terms of talking about, raising animals without microbiome, the differences in terms of like C-section births versus vaginal births, and that being how you would usually get your first microbes, and then what happens later in life in terms of particular disorders, particular diseases, particular like overprescription of antibiotics, which and of course a lot of substance abuse can create very distorted gut microbiomes, yeah.
I understand that it changes. Like if you want to drink a bunch of alcohol and you don't want to eat vegetables and fruits,
yeah,
your gut's not going to be happy with you, and it's going to be more acidic or something like that, just because of the sheer amount of alcohol and the nature of that substance.
Sure, but
it may be what it breaks down into and stuff like that.
Well, but it's important.
How does that translate to the brain?
Well, so one, it's these microbes that are doing a lot of that breaking down, right? So you're actually feeding them and using the metabolic, the byproducts of their metabolism in your own, as your own nutrients, right? So the species that you have are super important for what you can actually get from that food. Okay, so some of it comes from some of it comes from like actual nutritional problems, but again, like there's some focal studies. This is mostly animal work, right? Where they're controlling what bacteria that the microbiome of the animals, and then using particular kind of like psychiatric model systems for depression and fear responses and things like that to show the effects of microbiome and brain response.
So are they looking for correlations?
Say again, sorry.
Are they hopefully looking for correlations then? Like they know the biome is like this, and then they pick up an EEG state that is in some ways and maybe find a correlation?
Yeah. I wouldn't say so much EEG state, although that work is happening, and so the Mouse Imaging Center in Toronto, Sick Children's, has done some really great work on that kind of imaging. This is mostly behavioral in the sense that like you control microbiomes of the animals that are raised, and then you look at their at these kind of behavioral tests that are used as surrogates for depression, fear conditioning, things like that. Awesome,
that makes sense. So who said they were here working on dietary stuff?
Oh, so Bill is joining us from UC Davis Nutritional Lab, and
it makes a little more sense now that you broke it down. Thank you.
Yeah. So it's super interesting, and in psychiatry, this is assumed to be a really important, and a lot of this John Cryan's webinar focuses on the psychiatric applications, or the potential translational psychiatric applications of their microbiome work. There's also some discussion of like microbiome stuff is at a, I don't know if we want to say a peak, but is has a lot of hype with with it right now. And yeah, so I highly recommend Ed Yong's book as a good intro. Did sorry, did somebody find a link? Or I will.
I just want to mention some other stuff with the microbiome. It's really hard to, nail things down because you often find hundreds and hundreds of species, and each one changes a little up or a little down. So some of the work that we're doing, and we'll look at like a measure of the amount of hydrogen and methane produced by the fermentation of the bacteria, or some other small molecules from the bacteria that might appear in the urine or in the feces, and to get a broader measure of these changes, because when you look at it too detailed, it gets really hard to interpret.
What would you say? I mean, so Bastian from Open Humans was saying that there's a lot of like, if we were trying to look at our own microbiome, that there's a lot of variability in terms of, in our feces, like grabbing a sample is almost never a representative sample. Do you know much about that?
Oh, that's really, really true. We have to take many, many samples. It's so much work, especially when the assay costs is, hundreds and hundreds of dollars.
So we'll make plenty of jokes. Like, this is a real something job.
Yes. Yes. There was a guy who presented at quantitative the quantitative self meeting a couple years ago. It was very interesting. He did a bunch of antibiotics, and he had a colonoscopy where we flushed everything out. And what was amazing to him was he did kind of a before and after, and how quickly the original species recolonized, and he was trying to budge his microbiome into some other balance. But even after antibiotics and flushing everything out, things came back almost the same as before without too much time having to take. So there's two parts of.
Yeah, I mean that's the other interesting thing, and so I dropped a link in for Ed's book, and let me just check. Somebody might be asking on Slack if they can get in. Yeah, I'd love to know the kind of meetings where this kind of microbiome work is happening.
Very highly rated, 5 stars, over 600 reviews. Is this like a definitive text?
Oh yeah, I mean this is. I mean he's an awesome science communicator, right? And I see him being. I'm not exactly sure his background, but he's getting a lot of FaceTime in terms of like people asking about COVID response too. Anyway, but I do recommend it, and let me just put in the details for those, because after 6 the Meetup link doesn't, or people can't join the Meetup page. Okay. Oh yes. I did. Yeah. Sorry, Mateus. I got him there. So good. Well, John is not able to join us this evening. He has real work to do. And so we can't turn to John to say, hey, what's up with the Open BCI challenge and EEG notebooks? Jaden, would you have anything you would want to talk?
Yeah. I think, well, in regards to the Open BCI stuff, I think we're still just waiting for the IRB and everything, which means I think, so we're actually delaying the challenge a little bit. Is that right, Richard?
Yeah.
Yeah, okay. And then in regards to EEG notebooks, I'm waiting for John to just verify that a couple of these functions that I wrote work, such as like something to fetch all the data sets for all the operating systems, and then also the new analysis functions. And then once he verifies that those work and he's cool with them, I think we'll be ready to push out the new EEG notebooks and actually publicize it. So
sure.
That should be good to go tomorrow, theoretically.
And it looks like we want to make an email list. And
I
don't know what he was talking about, those weird websites, but it's very straightforward to make an email list using WordPress and maybe something like MailChimp. We already have a system in place at BCI we use MailChimp for our newsletter. Yeah. But it should be really easy with WordPress. And anybody that wants to make an account, just have a mail list. And that can even be separate from whoever wants to make a WordPress account.
And honestly, we should be able to get NTX social media or infrastructure to work on this. But that could be what he means when he's saying, like Abby could make that happen.
Yeah, if you want to make
Canadians are
a database to live somewhere that's more accessible to the Neurotech administration or whoever's taking care of that, then they can just relay that to me, and I'll smack it together with a plugin on the WordPress.
Yeah,
and that'll make like this. What that does is hopefully scrub the submission form where you put your email to make sure people don't abuse that.
Nope. Hello. So then the other thing Jaden was talking about there, for those who aren't familiar, is is just this like get data or this fetch function that so we were, all the Python neuroanalysis packages have this, and I don't know if NiLearn is necessarily the originator of those functions. Like M&E's got these functions too.
I think sklearn is where they adopted it from.
Well, and so M&E and NiLearn are all developed by the same people that brought us sklearn.
Oh, there you go.
They're all there in Paris. And it's usually core contributors, scikit-learn, that do these other packages. Okay. So is that something that?
I already wrote the function.
Oh, you got it.
Yeah, I just pushed it like a couple of hours ago.
Awesome.
Yeah, and so the issue is that the one that John had only worked for Linux and Mac because it used wget, and so I rewrote it using requests. And
gotcha.
It worked on Windows and it worked on Linux. So if someone wants to test it on Mac, that'd be great. But I'm pretty certain it'll work there too.
Gotcha. Windows.
Yeah, it's always Windows.
It's always. Yeah, at some point, can we just assume that Windows subsystem for Linux is installed?
Yes.
Maybe like next release or something.
Yeah.
Anyway, good deal.
But yeah, it should. I don't see any reason that we shouldn't be able to start releasing it, like telling people about it tomorrow. Just whenever John gets to checking these out, we should be good to go.
Gotcha. Well, I will say that so this week UC Davis also, different group at UC Davis, Stephen Luck and Emily, I forget Emily's last name, but as part of she's at San Diego State, did a webinar on this standardized measurement error. So it's a data quality metric for ERPs. So I think I sent you a link, Jaden, just to their original like May 1st blog post on this.
Yeah.
And it's, yeah, I mean, it's kind of what you would, it's kind of what you'd expect. There's definitely some nice built-in features if you use ERP Lab, which is an add-on to EEG Lab, and so of course this is all in MATLAB. Hopefully, I'd be really interested in working with anybody to bring this over for us too. And yeah, let me just.
Yeah, I could definitely take a look into. I found the link that you sent me.
Yeah, I mean it's.
I think Joe shared something called like ERP info.
Yes,
so that's hopefully push that out via social at Open Bci maybe. Yes, but it seemed cool. I liked it.
Yeah, so ERP info is Stephen Luck's website at UC Davis, and the ERP Bootcamp is how is like their blog name. I'm not sure what to call it. It's also their Twitter. It's also their Twitter handle. And that was what the webinar covered on Tuesday, I think. Tuesday, anyway, this week, and there is a link to that, which I will find.
Yeah, this doesn't look very hard to implement at all.
Yeah. It's just want to give people the YouTube. So you can catch the. Give me 1 second. I gotta go through his. Well, I'm going to drop in a link here, and yeah, so the talks are a little bit further down. Just 1 second, my daughter is knocking on the door. Okay, sorry about that. Yeah, so from the link, I Marimba lovers, good deal. Yeah, you can catch the data quality webinar from this week. And next week, as you can see from this, there's the ERP core, the discussion of the ERP core, which we covered at Hack Night a while ago now. I forget what, all the months are blending together. But this is an awesome, 6 experiment with 7 ERPs data set of 40 subjects. And, one of those is face scene N170 face object. I forget what they're using, but very kindly, they have shared their stimuli. So I'm definitely interested in having a variant of the, just one second.
I was going to jump in for just a second during the break. Is anybody familiar with, has anybody done some of the old school thought technology?
What would that be? Thought technology? Sorry?
Yeah, I was just during the break there, I was just asking anybody if anybody had done some work with the old school thought technology equipment from Canada.
Is that a company?
Yeah.
Do you have a link?
Let me pull that up.
Okay.
I'll put it on the chat.
And that would be helpful. So I'm going to see how I can share this. So I'm going to try and take notes here. So wanted to try and tackle the issue of us not having notes that from the recordings that people can catch links and things like that. I want to see how. So this is. So Kirsty Whitaker, who is also the lead on a autism project with Open Humans called Autistica, the Autistica platform. Anyway, she runs an online community around this Turing Way book for data science. and I want to figure out how do I'd love to be able to share our document the same way she does with a URL that is readable. So I'm still figuring that out. But if you want to join me in terms of keeping notes, and let me get some of these, getting some of these links into today's meeting notes. All right. So did you give us a link for thought? Okay, here we go.
Yeah.
All right. Okay, it wants cookies. All right. Electrical stimulation. Where am I hooking this up? Okay. I mean, I see a lot of typical like peripheral nerve. Oh, okay.
Yeah, a lot of biofeedback and neurofeedback.
So not familiar with them. You know, they look comparable to, say, you know, BioPack equipment, or you know, there's a lot of labs that still, you know, they'll collect all these kind of physiological measures and they won't collect EEG. Right. And you know, for a lot of clinical populations, that's enough and is important, you know. They'll usually, I mean, I would say I don't see them trying to get any eye tracking, but a lot of GSR, and certainly something on the homepage said something about stimulation. So, but this is the kind of stuff, I mean, you know, you can use Open Bci to collect a lot of these same measures. You know, what you're paying for there is, you know, potentially, I don't know if they're selling things clinically.
Yeah, a lot of neurofeedback people use their equipment.
Okay. Yeah. Yeah. And you know, and a lot of the equipment for brain stimulation, I mean, electrical brain stimulation, is still sold, is more sold directly. I mean, I'm thinking of like Fisher Wallace or some of those other companies, but yeah, there's a interesting open data set that was just talked about by, I think, Brain Products. Let me just show you this. So this is a really interesting. I think I've posted this on Slack before. My internet might be slow because my daughter is now streaming Bernstein Bears.
That's a good old show. Hopefully it's the old one, or is it a new one? Nope, froze up.
You can talk about neurotech and the development of human innovation, but against Bernstein Bears, no one can do anything.
Hey, Jaden, I sent you some music that might be more to your liking.
I'm checking out a little bit. I'm trying to update some of this documentation right now.
Sorry, my Ubuntu system just logged me out.
Classic Linux.
I'm not sure. I'm not sure what it was I said, but I was just pulling an open data set, EEG fMRI neurofeedback data set that, if anybody's interested in, looks very interesting to work with. Sorry for being gone. I'm going to slowly try and get my Ubuntu system back working. But let me just drop in this link. And okay, it is 7 o'clock. Not a bad time to just check in and see if there's anybody else that's joined us for the first time, is interested in introducing themselves and telling us a little bit about what brings you here. I try and reboot my Ubuntu system.
Well, let me mention something I'm got interested in recently. Has anybody been seeing some of the work on heart rate variability as a predictor of COVID-19?
Well, I mean, certainly. So to jump ahead a little bit to what I put in the Meetup agenda, a really, really interesting summer school this week was this USC Tides sensor-based, what was it like? Forget the full title was something like sensor-based SBB3 or SB3. I can't remember all the acronym, but it's basically like wearable ML for both neurological and psychiatric tracking. And so I was more interested in the psychiatric applications, but there was definitely some people focused on COVID and trying, and certainly all of them were collecting, doing like sensor fusion, right? So like using multiple data streams. So I could certainly see how heart rate variability would be, an important one. But you'd probably, like heart rate obviously by itself could also be a number of other things, right?
Yeah. Was this a news story or published paper?
So give me 1 second while. Yes, I would like to.
I can understand that because I did see a news story that was indicative of many doctors' opinions, which said that it affects more than just the lungs. It's not just a lung disorder; it is affecting multiple systems.
Absolutely, yeah. and yeah. So there's this was a cool summer school focused on all sorts of devices that are picking up all different kinds of signals from you, right? So some of them were focused on just using your audio mic, right? So could you do, how much diagnosis can you do a COVID just with hearing somebody breathe? Right?
Yeah.
And
you can feed that to a neural network, might work.
Yeah. Right. So this is what this Tides. So give me.
And honestly, from what I've learned from asking many questions of medical professionals and things like that, I don't have a degree myself, but you say cardiopulmonary system, they're inextricably linked, the heart rate and respiration. The lungs need to get every bit of oxygen on every single breath so that the heart can continue to do its work. And I would think that is going to inextricably be linked to heart rate variability if the lungs are functioning poorly.
Yeah, and even just breathing rate, breaths per minute will change heart rate variability.
Yeah, and then you've got people, I've never met anyone with COVID. I haven't treated anybody, but when you can't breathe as well, I do have asthma, so I can say that. You end up doing more breaths per minute because you're not getting as much oxygen per breath. Yeah. And then you have the risk of hyperventilating, and then you're just going to pass out.
Yes.
It's a whole. That's why I'm not surprised that the coronavirus is the disease, but it causes this massive syndrome.
So I'm just dropping in a link here to the summer school this week. Okay, and yeah, I have on my other computer, I have all these tabs with relevant papers that I was hoping we would talk about. But if you check out check out in particular the speakers, and so if you jump down to the set of speakers, and let me. So some of this, and I'm going to just pull up someone's work to start talking about. This is Clemens Stockel. Not sure how you'd. pronounce it, but this, so this is a super recent paper, and PNAS. So predicting personality from patterns of behavior collected with smartphones, right? So this has been, this is obviously, I'm giving, this is like an extreme example, just being like, big tech's favorite thing is like how much information, or, how big a kind of like, not diagnosis necessarily, but, how specific or how important a label can we give you on the basis of, how you use your phone, and like how much, but remembering that like your phone has got an accelerometer, it's got your, search history, it's got a microphone, like it is a compute platform, right, with a lot of different sensors. And anyway, like a great example where he's using the big five personality inventory, trying to connect that with your phone behavior. But let me just see if I've managed to, I guess Ubuntu's not going to tell me what went wrong. Anyway, I want to thank Jyoti Mishra for telling me about this kind of last minute, but super glad that she did, because very, very relevant. I mean, it could have easily been I mean, it's the kind of hardware or wearables, is the kind of wearables event that I would hope that NeurotechX could do similarly, there wasn't a lot of EEG in this. This wasn't focused on those kinds of measures, but it was yeah. But I'm going to pick out a couple people from there in terms of highlighting their work, and Jyoti's obviously is probably the most relevant. Certainly, she's the one formerly Adam Ghazali's lab, who was really focused on with others, but focused on can we get wireless systems into people's houses to be also collecting data at home, and in more naturalistic settings. But she's got a lot of so that all the talks will be available on YouTube soon. Unfortunately, I don't think they're up yet.
I think it said a couple of the slides are up.
Okay, so the slides might be enough. Here, let me just pull this up on. Well, here, let me. One second. I'm going to try and rejoin.
You can access most of the videos on the web from the website.
Okay, great. Whoa, sorry.
Using like the MP4 link.
Hello. Sorry. That's great. So I highly recommend it. Catch what you can. Certainly make sure that Jyoti's is one of them. Really interesting work. I'm really hoping that we can get her or some people in her lab, especially some of the engineers, talking about that I know she's got a closed loop, I think it's like a Simulink framework for doing closed loop stuff. But in particular, you know, this was particularly relevant just because, again, like I've been catching the Open Humans community call, and you know, as part of Open Flu and some of the other related projects, I don't think Open Humans is doing the COVID tracking, but you know, Open Flu is somewhat related, and certainly is doing that kind of mix of wearable sensors that you'd need to be able to distinguish, you know, COVID from flu, for instance, right? And here, let me just get back. Don't. I seem to have lost my Google history here. Let me. Okay. Yeah, Tiago Falk was also great. In terms of tracking a whole bunch of things that we would. Okay, let's see. So this is. Yeah, so here's Jyoti's slides, and you know, a perfect application of, you know, if they don't mind me calling it neurotech, and there's you know, Adams and Tim Mullins' glass brain. Yeah. So, another interesting development in terms of this kind of neurotech. Apple. So if everybody remembers Julie Brown from MindX, and she was talking about their Apple Health Kit project, which I think was related to Epilogue, but it was like, you know, tracking with your watch for, you know, a neurological disorder, right? News yesterday is that Apple's funding a small study in the sense of like, I think the first rollout is to 300 people, but a small depression study at UCLA. And I think I know the lab that might be involved with that. But what I thought was interesting was not only is this Apple, you know, like this is definitely somewhat following Clemens Stockel's, you know, predicting from patterns of behavior collected with smartphones, right? But add Apple Watch, and you know the other you know news-ish rumor is that Apple Watch 6, I think it would be, is going to have a pulse ox.
Yeah, they can do it. They just need to change the type of sensor that they're using.
Yeah,
we learned that.
Yeah.
At Open Bci from working with Sean Montgomery, and he's also working on Emotabit, and they can do pulse ox there too. And he was just explaining it's like a different color light, maybe, and I guess it works slightly different.
Sure. Well, so I mean, yeah, I mean, this is certainly what we're trying to do, or you know, that's Edgeduino is trying to do. I mean, together with Josh, we've posted some things looking at, you know, Adafruit had quite a few pulse ox devices in the recent weeks, and it's show and tell, and I forget the show she does where she's just showing off particular things.
Are these for sale now on Adafruit?
Yeah.
Let me go check that out.
But so anyway, it was just interesting that Apple was doing this depression. You know, it seems to be funding a depression study using HealthKit, and apparently the rollout is going to include, they've got a sleep device. So I had to look this up, and I'm afraid I've lost all my open tabs. So, but Apple, on the Apple Store, there is a sleep device that begins with a B. It's like, the Bedit sleep monitor.
Where's it worn?
Is
it non-wearable?
It's not a wearable. It's like here. Let me just I'll drop this link in.
I know I've seen. I think at least one product may have been
actually
from a commercial where you like put something under your mattress, maybe.
Yeah. So apparently Apple bought Bedit in like 2017, and they haven't made the company disappear. They're still selling devices, and a part of this depression study and is giving people one of these as well as you know, your standard Apple devices. So they're definitely trying to roll in sleep, which.
And I've even bought, I think, a sleep tracker. I may have paid for the app, and they can pretty much predict your sleep cycles just on how you're moving in the bed. And so it looks like maybe this is just a very fancy motion sensor, a lot better than what a phone on a bed could do.
Yeah. So let me just. So there are some things that you can't get from just activity trackers. And let me, sorry, I've got to find everything again here. But so at Monday's NeuroHack Paris, and Let me just remember her name. Yeah. Muna, right.
Yeah, I see here that Adafruit is just selling straight up real pulse ox.
Yeah,
for 30 and 50 bucks.
Yeah,
and
they had,
and it says limit one per order. That's what I wanted to say. Max one per order.
Right.
Because people, I guess, would be panic buying this, doing nasty stuff.
Yeah. I mean, and that was, that was what Lady Ada was talking about, why they were focused on these devices in the first place. Just 2 weeks ago at NeurotechX Paris, we had, let me remember, we had the CEO of Muna join, and Colleen June, talking about their sleep device. And it is also a pad, but it's a pad that that is somewhat more focused on catching your temperature sleep cycle. So it's doing, the activity tracking that is that's similar to what's that company Nokia bought? Withings. So Withings made a sleep pad too that reminds me of Bedit. But what Muna was adding was definitely trying to catch your temperature change, right?
Are they like getting the BTUs that end up being transferred to the pillow?
Yeah. I mean,
you're right. They know that you're right on the other side of the pillow, and there's only one place for the heat to go.
They're, I don't have one.
Are they predicting body temperature, or just like we know you're hotter at that point of the night?
No, they're definitely trying to catch, data related to your body temperature, and then they are potentially trying to do something about it. And now this is these devices are available for sale, but these are also active research products, in the sense that, like, they're working with sleep labs to try and figure out, if we knew your temperature, what should we do about it, and how does your thermoregulation relate to the quality of sleep?
$400 though.
I don't. Well,
I mean, I'm sure it probably works. It for everybody else that may not have looked at it, or if you're looking at this later, it looks like it forces air into the pillow to keep an always cool pillow, and that's what they're selling.
Yeah,
but it's cool to know they're trying to do some cooler stuff in the background.
Let me. So it's interesting. So we were going through the Candel book on Candel Neuroscience, and its chapter on sleep. And anyway, there's one of the sleep disorders that they talked about is people who obsess about trying to improve the quality of their sleep. So I mean, I do think it's a big unknown of how well, like, even if let's say you had better temperature sensors, like taped into the inside of your armpit or something like that, right? How would you act on that? Like that's very unclear. I also asked if they had done any work with sleep labs that were also collecting EEG, and polysomnography. I mean, I love polysomnography because it's so multimodal, right? Or it's because it's collecting data from so many sensors, and so it's very much in the spirit of this Tides summer school. What was it called? Sensor-based behavioral machine learning. Right, where, you know, certainly one thing is just getting your sleep, you know, what stage of sleep are you in, right? And you know, that's a measurement that needs to be done with EEG, EMG, and EOG, right? Like you need all of those, and certainly, especially when you want to look at disorders related to those, right? So it's like, is your EEG and your EOG saying you're in REM, but your muscle tone hasn't changed, right? That would be of interest, or, you know, anyway, there's yeah. So Muna was taking it in a direction that I hadn't seen anybody try to get temperature before. And let me just. So since we are talking about sleep states, there is a new paper.
I just got to say before we move on, if the next Apple Watch really does have blood oxygen, I mean, you can straight up give somebody a warning that they need to go to the ER. That's so easy to detect. And it's more power. They had to wait on the, I can't remember who they collabed with, maybe like Carnegie Mellon or Stanford. I think one of them was Stanford, but they did the HRV study so that they could predict, you know, give people a warning now, and that's already in place. But blood oxygen, if you've got that metric and it drops below 90, you need to see a doctor. So easy. And it could really save more lives than probably their current metric for HRV does.
Yeah, I mean, it'd be great, right? And so you'll see this in let me just see where we've got
I'm actually like a little bit scared now that maybe for times when I'm not paying attention and my O2 drops, it would make me more aware of that. It's different. It's part of the quantified self, learning more about what we do during the day and why we do it.
Sure. So I just wanted to put on this particular slide from Jody's talk, where she's talking, you know, like this is, you know, very much like what we would want to do with, say, Open Humans, right? Is catching via, say, you know, an iPad or iPhone app, catching, you know, some sort of depression inventory or, you know, psych inventory. Also, you know, during the day, being able to put on just a consumer EEG hardware that you know, person can apply themselves to get some brain measurements using, you know, Fitbits or other things. You know, your watch, potentially your phone, and then the diet tracking and the mood and attention tracking, both a little bit more complicated, but I love, or, like this is again, like so close to the, multiple data streams that you have available, or, people are tracking quite well with Open Humans and other kind of quantified self groups. Anyway, I love this.
There was a diet tracking app that I tried out since that was in the slide, and it helped me lose some weight, maybe temporarily at a time in my life, and I was able to say, oh, I didn't exercise, but it seemed to have all of the food that I was eating already there, and I just had to search it and add it, right? And come to find out, there was a data breach involved in that company. So I just went into that app and I was like, I don't care anymore, it's gone.
Yeah.
But I don't think my data was being sent off. I think I was doing everything right on my local device. And I just really, if I was going to do a dieting app this time, I'd want to pay a one-time fee, not a subscription, and I want all the data to be on my phone, nowhere else.
Sure.
To prevent things like that.
Sure. I think Open Humans is doing a good job, or at least more transparent than any particular commercial company in terms of what they're doing with your data. Unfortunately, I wonder if my work on my spectrogram, I've wanted to get some feedback in terms of the description I've written up for Open Humans My Spectrogram, where we can just upload, resting EEG and get your parameterized FOOF fit, something like My Connectome, which was another, not everybody focuses on the other measures that My Connectome was getting, because he was getting MRI data every day. Not everybody focused on all the different other phenotype measures that he was collecting on a daily basis. And, those are just as important, especially when you're trying to do a model-based analysis of that. Anyway, still writing that up. And yeah, anybody who's interested in my spectrogram, please get in touch. So 7:30. Let me just check. And any I'm just checking to see if everybody made it in. Okay. So I'm going to pull up.
So Morgan, I just want to mention for a minute.
Yeah.
Thank you. I'm going to have to go in just a couple minutes, but
sure.
Man, the stuff from that summer school is really a gold mine. Yeah. So I'm really looking forward to digging through there and some of these other links, and looking forward to the next meeting. That's,
please. And yeah, I'd love to, again, with the demise of Ubiome, it's a real question for me, and I don't know if anybody in your lab can speak to, you know, are there consumer kits that we can still get? Are there any differences between them? You
know that's a really good question, and I've been asking some other people in our lab those same questions. I haven't gotten good answers yet, but I'm going to continue to ask that, because I myself was ready to take the plunge with the microbiome, ubiome kit, and then then stopped, and now I'm looking for another place. So
yeah. So I'd love recommendations for other places. I think that Bastian's comments about you know, the variability in terms of just taking a small fecal sample and like just not being representative is a huge issue.
Yeah, it is. You know, I'll take that. You know, we've debated that around a bit, you know, on this small sample, and I'll ask one of our senior scientists kind of where they're at with that. You know, what we can do to get around that.
Please. Yeah, and then you know, if you want to, I mean, we're kind of booked up in August, but I'd love to come back to the how do we track stress, and you know, these other, I mean, some of John Cryan's talks about you know, nutritional supplements and their effects and on your microbiome and brain effects. Yeah.
Well, I have a colleague who he could really present pretty well on carbohydrate and stress. So yeah, that would be good. Maybe sometime in September.
Yeah, that'd be great. Thanks.
All right, well, gotta go, but thank you very much. It was very, very good.
Take care. Yeah. We'll try and. Are you on Slack?
I haven't got everything all settled up yet, but I'll try.
Okay.
Try to get.
I can send you a link.
Oh, that'd be great.
And you know, the other thing, I mean, I don't know whether it would be easier for you to use Discord, but you know, just in terms of staying in touch with people, you know, Slack is our main platform. And so you don't have to wait for another week. But I'll send you a link via Meetup. Okay.
All right. That sounds good. Thanks. Appreciate it.
Take care.
All right. Bye-bye.
Yeah, before we move on, is there anybody else who had one who would like to introduce themselves, and two, if you've got any questions that any burning questions for this evening, please let me know. So I'm just going to pull up another author from the summer school.
I saw something cool on LinkedIn. It was Colin Fosnot and another guy, and they called themselves the BCI guys, and they were breaking down a Neuralink claim that Neuralink could put brain straight to your brain, and they brought it into reality very quickly, which says we already have this. It's called a what do you call it? A cochlear implant, and it has, the best ones have, they said, 22 electrodes, and it currently sounds like, and I guess they would have somebody that lost their hearing. Maybe they had perfect hearing at one point, so they know what good music sounds like on good headphones. But they said it sounded like real tinny, and it's not all there, and you're really not going to get, I don't know how you reproduce the low end sensation of like your body rumbling with a subwoofer, and how that goes in through the ear canal to the eardrum.
Yeah, I mean it's
That's really hard to reproduce because some systems even now have a subwoofer that's like on your back or part of the chair, just so you feel it.
I mean, I don't think we have to point out that Musk says a lot of things that he's not ready to.
That's what they said.
And so it's kind of, that anyway, plenty of people have demonstrated that in multiple domains.
So the summary of the video, just to save everybody a little time, was that yes, we can do it. Yes, it's theoretically possible because they said all of the existing images seems to go over them more. Yes, baby. All the existing images say they're working with this high density invasive system over the motor cortex, and you could theoretically put that over the audio processing that's near each ear a little bit. But then again, it's possible, but it might not sound better than regular headphones ever, or it might not be perceptibly better. It might actually even be worse.
Yeah,
and we've just evolved these wonderful ears over thousands and thousands of years, and they work beautifully. And
yeah,
it's cool enough that we have cochlear implants at the current time.
Right, and those are really super impressive. And how much we've worked out of the auditory pathways. So somebody was asking about auditory EPs or resources for auditory EPs, and just a quick search reminded me that this is one of the earliest tests that we do, brain function tests that we do, is auditory brainstem, and it gives us this measure of how well the hearing system is working before a child is necessarily verbal, right?
Actually, I mean, there's been conclusive research to show that playing Mozart for babies, can be a positive influence, and that if that's how far they've gone with it, that means fetuses can hear okay. They can hear something.
Well, we know that. I mean, certainly in developmental psych, there's the famous study of I forget if it's like just outside of Tokyo, but it was mothers on the flight path to the major airport, and they were basically looking at language delays due to the noise drowning out, the being able to hear the language you're going to learn. But
you're talking about the fetus's time spent in an aircraft?
No. Fetus's time spent in a noisy environment.
Oh, just a noisy environment.
They couldn't hear just the sound cues through the belly. And
okay, are you talking like mothers that fly like once a month?
No. I don't know. So when where I grew up in London was underneath. It was like, yeah, it was really
living next to the public transit
to live, and so we were under the flight path to Heathrow. And I remember I've taken the people there before, and they'd heard what I'd told them about this, and then when we actually got there, and then they were like, "This is just all like after like the fifth plane has flown over your head."
But you have to like secure everything to the wall with nails because it's going to shake the house.
Like, wait, did this just never stop?
Oh goodness!
Yeah, it's the busiest airport in the world. Anyway, but this is a study that was trying. What they were trying to get at was are there cues that the fetus is picking up of language, before they're born, anyway, that I don't know how well, it's been a long time since my developmental psych, but that's one study that I remember relates to that particular.
That's so hard. I don't know if they could ever measure that. I mean, can they do EEG on a fetus currently?
So
it sounds like you'd have to have some really good reasons to do that too.
Well, but they do, right? So there are those kinds of cases, so there's a couple of things, so one, it's a super active area just in terms of imaging babies, imaging fetuses, but they're actually specialized MEGs that are designed for looking, because the magnetic field is, it's more about the number of sensors, really, and but there are specialized MEGs for this kind of fetal brain signal detection, and you can imagine what they're trying to do. I forget who's.
Is there a clinical reason that you can think of where they would order that?
Yeah, where they're worried about the fetus.
Oh, they're just trying to see, is the brain functioning normal, I guess.
They're worried about the fetus and the mother.
I mean, just like I said, that sounds very serious.
Yeah. But I'll just give you a couple.
I mean, but it'd be good to use those machines for just some research on the side if somebody's got it. Get some healthy babies in there and healthy moms and see what it's like.
Yeah. I mean, it's interesting, right? So here's a particular paper. Got some of my favorite people, Fabrice Wendling from Rennes, and who's done some awesome finite element modeling, mostly focused on epilepsy, but anyway, he's on here, as well as Rafini from StarLab. And StarLab's also gave us the wireless EEG TES systems. Anyway, just talking about some of these, some of the challenges here that, for anybody who's interested, what I was actually trying to look for was who's making the particular scanners. Because there's some great pictures though. Wow. MRI is pretty hard to do as well on fetuses. But let me just share this particular picture. Apparently, this system is capable of recording 64 channels of simultaneous EEG. It's funny that they call it FMEG, which reminds me of fMRI, but they mean fetal MEG. Anyway, so.
Oh my goodness, that is a special machine.
Isn't that?
It's like no other reason for that.
So.
Yeah, cortical MEG is what everybody's used to seeing.
Yeah, anyway, I wanted to just keep focus on some of the Tile Summer School speakers, and so this Tiago Falk who's, I believe, in Montreal. Let me see if. And of course, I'm not going to get. Anyway, looking at. Let me just see if. Let me see if we can get his slides. Ooh. Okay, he's got a pre-recorded presentation. Yeah, so it's the University of Quebec and the Musée Lab, Multimodal Signal Analysis and Enhancement Lab. So very much focused on, you know, stress, anxiety, fatigue, and I think, and yeah, and a description of a newly developed body-computer interface-equipped virtual reality headset for user-aware serious gaming. So that Tiago's is another one of the talks that I'd highly recommend. And yeah. Let me. Hello?
So it says, okay, baby.
Yep, go ahead. Are you talking about Tiago's, our Musée Lab? Let me find.
Yeah, sorry, my he wants to play a video game. He can't play by himself. So I've got to help him. But it says like first responders and things like that. Sure. So if you really could have a commercial application for that, are you going to tell your EMT, like, don't send that, don't send those two EMTs out on that call because they're too stressed from that last call, and send a different one? I mean, you can't, I don't think you can do that from an insurance standpoint. You have to send the closest unit. But what are they going to do about it if a EMT is upset because they just watch somebody die?
Sure.
I mean, they're going to be stressed.
You know, but that would be great. That would be great information to have if you had the option of sending different people, right? And certainly,
I think existing supervisors have to be doing that. They have to say, you know what, y'all go do a hospital transport or something like that to finish your shift. I would think they're considering that human factor, and that these aren't just robots. And in fact, I've seen a lot of reports saying that in the future we really shouldn't be replacing hands-on medical care with robots. I mean, it's kind of being done in COVID. Some people probably are against it, but there is just something special when a human is taking care of a human and making sure that they're cared for and alive. A robot just takes the humanity out of it.
Right. I don't think anybody's, you know, I mean, certainly none of this is speaking to that particular question. But you know, the neurotech related question is, can we provide insight? You know, can physiological signals give you an actual measure of stress, fatigue? You know, certainly, you know, I mean, one of the probably biggest areas of military research has focused in on just when should somebody just take a break, you know, and especially if they're being used for signal detection, So like radar operators or things like that. You know, it's one thing to have a supervisor. It's another thing to know, you know, how that person's doing.
Yeah, there was a movie I watched at least where, you know, it's an aircraft traffic controller and he makes a mistake, and then the movie is like his response to that in his life.
Right, right.
And it would have bbeen great in that scenario to say that, you know, you're overstressed. I don't know. The other guy was like in the bathroom or something. and he tried to do 2 control panels at the same time.
Sure. Well, so I really love. Let me see if I can get a link to Muse Lab. So some nice papers there, getting at this. That I've dropped in the chat. And here's a link for Muse Lab. Let's see what they've got in terms of. Here. So I'm just on their software page. They got a MATLAB toolbox for speech to reverberation modulation. So I think. Okay, and that's a normalized. This has something to do with cochlear implants too. An EEG server. Look at this. Mules. An EEG acquisition streaming server that aims at creating a standard interface. Look at that.
Wait a second.
NoBio, Muse.
Where does this run on the web?
Oh, look, it's Hubert. So I didn't realize Hubert is related to this lab. All right. Well, now we're gonna have to. Okay, so I just went to the GitHub page, but I don't see my. Okay.
Are you kidding me? Is this what I think it is? So we could run this on a server. Let's just say spin it up as needed on AWS, right?
Right.
And then they say you can send it over TCP/IP. We could get 10 people's EEG time locked to 10 different to the same video at the same time.
Wait. And this is lab what?
I don't know how they're connecting to Open Bci though.
So this is using LabVIEW, which can't be the whole story here. So
what does this run? Is it like a how did they make it?
LabVIEW is a platform. I mean, it's usually a platform for controlling data acquisition cards and potentially other specialized hardware. I get the feeling that they must be doing something more with it than that, because given its support of so many other, like they're saying, like a LabVIEW emotive toolkit, a LabVIEW Open Bci toolkit, a NeuroSky LabVIEW driver, and a LabVIEW.
So this hasn't been updated recently, so they're not using BrainFlow. They must be using.
No, they're
I don't know, direct You get, if you really want to with the Cyton, it's just a serial port. You can read from the serial port any way you like.
I think it's something about this BioHarness 3. Sorry for my dogs. I'm not even sure if this. Oh, here's. Yeah, I mean, they were obviously using LabVIEW to interface with multiple devices via Bluetooth, right? And if they were going to redo it now, they would be using, you know, LSL. Let me just close the door, sorry. But
yeah, unfortunately, looking at these .vi files isn't giving me any insights into what they're doing in acquisition.
I can tell you. So, I mean, I built LabVIEW systems back in like the early 90s. I mean, I know it's still used today, but
so was this run in a lab, and they're sending from different computers to one central computer on the same local network, or were they literally trying to go over the internet?
No. They were just this was local. This was
okay.
Local. And
okay, so not quite the holy grail of what I was thinking.
No, I mean, look, you can tell that they're doing, this is like early. This is just like pre-LSL. But it's super interesting because you've got Ray Cassini, you got Hubert Banville, and this PI Diego Falk on this, you know, 2015 paper. And so for yeah, I mean I love all the hardware that they support.
How did you get to this paper?
So this is off of
I'm on the software page.
GitHub. This is off of the GitHub.
Okay.
And apparently there is some Python to this.
Yes, I saw that.
But like you said, like this is not a maintained project. But super interesting because if for those who didn't catch it, I dropped in a link. So there's the most recent archive link that I dropped into the chat is the Uncovering the Structure of Clinical EEG Signals with Self-Supervised Learning. And so this is a recent paper on archive that that I posted in Slack a couple days ago. And I was saying like, hey, look, this is like an awesome group of people. Because, you know, the last author is Alex Gramfor. You got Denny Engman. And for those who don't know, Apo Hyvarinen is the developer, is the Finnish developer of Fast ICA. So he's back at the old Helsinki University of Technology, you know, like in the 90s was doing awesome things like Fast ICA. And anyway, but Hubert Banville is the first author on this paper. And so he's at Paris-Saclay and Interaxon. Anyway, this particular archive paper is awesome in that they're using the TUH, you know, they're using the Temple University Hospital data set. And yeah, So demonstrating a promising technique for discovering structure in unlabeled data to learn representations of EEG signals. And so, I mean, I think.
Oh, wow, that sounds super applicable to everything.
Yeah.
My goodness.
Yeah. So
can you post that? Where's that at?
If you go up in the chat, it's the last archive paper that I dropped in chat. And
wait, what do you mean archive? Oh, okay.
Yeah.
Okay.
You see it? And so super interesting because they're doing two things. One is sleep scoring or sleep staging, as they call it. So that's making, taking the EEG and producing a hypnogram, right? So just what stage of sleep are you in? And then pathology detection. And pathology detection was again like the focus of Tonio Ball's paper from like his March paper and archive that is focused on EEG pathology detection using brain decode. And I want to say that Robin is one of the first authors on it. Give me 1 second.
Well, I've got a browser full of links at this point, but I think we're gonna have to start winding down for bed. So here, baby, come say goodbye.
Totally.
So say goodbye, baby. Oh, bye-bye.
Bye.
Good night,
bye-bye, everyone. Take care, y'all.
Take care. I dropped in a one more link, just relates to EEG pathology from the brain decode group. Anyway,
sweet. I got it loaded. All right, take care.
Night. So this particular paper, okay, it goes back. It's February. You can see Robin Schurmeister, who spoke at the BCI 101 on brain decode, and obviously Tonio Ball is the last author, but Frank Hutter is the developer of AutoML. And yeah, so super interesting that the M&E people are looking at this, and is Apo Hyvernanen is like super famous in signal processing. So that's already really interesting. And yeah, and they're not doing anyway, it hopefully, I mean, we focused on this paper for the Paris meetup this week, just because we were joined by somebody from Paris-Saclay. But
I might actually send someone over to the Paris meetup.
Okay.
Yeah, this girl up from France randomly DM'd me on Twitter. I guess she saw that I like interact with Open Bci a few times.
Sure.
And she's interested in getting like a headset and wanted to know some questions. So
cool.
I figured this one's probably not in her time zone to hit up.
Gotcha. Yeah, awesome. Yeah. Please. You can always point them to meetup.com/coglab.
Oh, that's right, because they run the Paris one, right?
Yeah.
Cool.
That's how because I think Coglab predated NeurotechX, but that was great. I mean, so we were joined by a physicist from Paris-Saclay from one of the synchrotron labs, but I had this wonderful example, and of course the really cool surprising bit was that is the first author.
Yeah.
So when we were talking about him spending time between Paris and Toronto, I didn't realize that this is who we were talking about.
Yeah. That's funny.
So super interesting. I didn't want to stop. I mean, certainly Diego Falk's lab. I don't think, none of this LabVIEW stuff is anything I wanted to try and get working again. But super interesting applications. Let me, oh yeah. So I just wanted to talk about one more. Oh, well, we've got plenty of time.
Yeah, I've actually got to get going for the night.
Sure.
Oh, let's see you later. Talk to you tomorrow.
Okay, talk to you. Yeah, good night.
Good night.
So just dropping in another paper from this time from Bob Knight's group at Berkeley, relating to electrophysiological marker of arousal. And they're looking at both, basically this is extending, Bob Knight's group's focus on, noise as a marker. Noise is information. In particular, the changes in noise as they relate to sleep and other. Well, in this particular case, this is just sleep versus wake, and but this is, as some might be aware, since we've talked a lot about Foof and Bradley Wojtek's work, especially that most recent paper where they were focused on the 1/f slope as it relates to a lot of that combo paper that they did with Allen Institute data, that was terrific, and talking about looking at noise up above 100 hertz. Anyway, this very much relates to that in terms of getting at that drop off. You get a faster drop off, and therefore steeper slope in the I think it's REM. Oh, and also propofol anesthesia. Okay, yeah, it wasn't just I think some of the. Let me just jump ahead to the sorry, I can share this. So this is the 1/f during the propofol, and yeah, so these would be your typical Bob Knight patients that are in for probably epilepsy surgery, and can I make this bigger? Cool. Okay. Yeah. So here, let's see if we can. Right. So anesthesia is in blue, and yeah, you're seeing that steeper slope. Oh, they have EEG and, oh, that's interesting. They've got EEG and intracranial. Yeah. So, but in both cases, yeah, this is a significant enough difference in the slope. Is this the range? I'm trying to, I don't know if you, can you see my, yeah. It's, I think they're only looking at the slope in this range, but I can't. Certainly the earlier paper, you know, Bradley Wojtek's papers were always focused in very high gamma range. And let me just see here. See if we can get a similar picture for them. Okay, so yeah, so rest is in so here at the top, this is what we call a hypnogram. Just relating to your particular state, sleep state. And yeah, so your stage 3 sleep and your REM sleep are both having this drop, much steeper slope, and in intracranial, it doesn't seem so clear. Let's see if they say anything about that. So, okay, they're saying study 3, they got polysomnography recordings from 20 healthy subjects. I mean, it doesn't look like it's saying it's still significant. Okay, guess it is. Okay, so study 3 is healthy. Okay, yeah, they've just got EEG in those. And is that the same for? Okay, so they did EEG, I think, in the same patients in this study 1. Oh, no, study 1 is orthopedic surgery. Okay. Still super interesting. And let's see if they've got anything else on this. So I don't know if anybody else had followed the discussion that had with Kalar about EEG versus fMRI in terms of wake sleep discrimination, or, you know, certainly like the kinds of how clear fMRI differences would be in sleep versus rest, or wakeful. But this adds to the story in terms of what differences you're seeing in the noise itself. And this is, let's see if they do. I don't think they're using FOOF. Let's see if they say somewhere here. Yeah, please. I'm not hearing you. Sorry, is somebody trying to speak? It's very broken up. Well, feel free to chime in if you've or drop any question or comments in the chat. My dog is getting antsy for some reason, and let me just let the dog. There was something going on out there. Yeah. So let me just check what other items I put on. Oh, well, so I wanted to cover the let's see. So could have a look at the EG notebooks documentation, and in particular, actually, would like to look at actual code of the experiments. Okay. Jaden has updated the big refactor 30 minutes ago. glad Jaden was getting stuff done during Hack Night. So let me. All right. So I'm having a look right now at code for the N170 experiment, because what I'm looking what I'd like to do is get, so let me just remind myself of her name. Oh yeah, Emily Kappenman. Oh, okay. Jackie's got a comment, which is, I'm sure, a great one, but I have to copy and paste it somewhere bigger. So my old eyes can read. Okay. I'm sorry your audio is not working. But, oh, they did use, oh, okay. Okay. Cool. Good deal. It, let me, where did I let that go? Oh yeah, here we go. Why is it jumped? Where's methods? Yep. Okay. Yes. So Matt Walker's on this paper. I see. Okay. This is coming out from the sleep lab at Berkeley. Aha. So what do we got here? Now, interesting relationship of slow waves and the spectral slope. Particularly relevant for the. Oh.
Baba,
yeah.
Can you put one Sophia Princess show?
Can you ask Mama?
Well, it didn't work.
Do you tell her she needs to use the other remote?
Okay.
I'm just having a look at this other. Trying to see what. So I know that they're using these segments of N3. Okay, so this is definitely from the Matt Walker component. So this is healthy controls. What are they doing here? Anyway, glad to know that.
Didn't work.
Sorry, I will be right back. Just one sec. Don't know if anybody jumped in. Oh, don't apologize. Would you you want to catch up? So sorry, I was just helping my daughter with a show while we're talking. Yeah, so check out the Divesh. Just check out the chat, which hopefully, let me know. Can you see everything that's in our chat? Good. Okay. So yeah, the last paper that you see there, the Life Sciences paper, is what I was just talking about. And let me just quickly share that again. So I was just talking about this particular paper that focuses again on, I mean, they're talking about this electrophysiological marker of arousal, and what they're specifically referring to is a change in the 1/f slope that you see in, for instance, right here. So in this case, this is 9 subjects that were undergoing anesthesia for non-brain related surgery, and they've got what looks like, I guess, 20 channel EEG, and then this is their slope measurements in terms of wake and anesthesia, and the significant increase in the slope related to, you know, a loss of consciousness, or however you want to describe that. Let me just see what these particular, I guess this is well, and then in this study too is intracranial patients, or, you know, intracranial electrodes in patients undergoing pre-surgical planet mapping for epilepsy, and they're seeing all this, a similar slope difference. Anyway, that was sort of what we were getting at, and it's kind of the end of the evening, and but if you've got any, if anybody has any particular issues or things that they wanted to talk about, I wanted to get at, because I know that we'll be talking about this next week. So up at the top of the chat, you can also see, let me see where it is, but the ERPinfo.org virtual bootcamp is another great resource from UC Davis that's again focused on event-related potentials. They've made a lot of educational material available under Creative Commons license, and next week's webinar, which I believe is August 12th. If anybody's got that particular and I'm not seeing it right now. Oh, yeah, right, here we go. Yeah, it's August 12th. Sign up for that if you can. It's usually, I think it'll be the same time, which is 8 a.m. Pacific time. But if you can't make that, there's they post it on YouTube pretty soon after. And anyway, that's going to be. And you can sign up for it here. Core shares, so some of our goals with the Open BCI challenge are very, very similar to Core in terms of a goal, which is to collect, in as short a time as possible, to get good ERPs, good canonical ERPs from multiple paradigms, and some of the particular paradigms that EEG Notebooks has already implemented are those same ones, which at least the two that are the closest to ERP core experiments are the N170 and the mismatch negativity. So what I thought I could do with some of the remaining time was get into, so Emily Kapanen has shared the stimuli that they're using. So ERP Core makes all their experiments available for Windows because they're using presentation, which I believe was actually developed out in UC Davis. So that's the presentation, which is a hard app to search for online. But if you check neurobehavioral systems presentation, is the neurobehavioral systems is the company, and so it's a Windows only app. But anyway, she sent me the stimuli, so we could use those same images, and synchronize with their experiment better, just in terms of stimulus presentation time, interstimulus interval, things like that. And the nice thing being that in addition to making their experiments available, they have also released 40 subjects worth of data using essentially a 32-channel EEG system. And that is also perfect for us because, when we're using these particular, consumer devices or, Open BCI devices, we now have, kind of like research grade ERPs that collected with essentially the same experiment. So it makes it a little bit harder just because their particular setup hard for us. They're using a biosemi active electrode system to collect data, which also has a somewhat non-standard reference, and not exactly. Yeah, it's not something that we can, certainly with the Muse, it's not something we can recreate. But what I'm certainly interested in doing is, like, we should be able to use their data to say what we should be getting with a Muse if we re-reference their data to the way that Muse is collecting data, for instance. And therefore by using essentially the same stimuli and the same timing and et cetera, we should be able to develop a template for what we should be seeing. And there are some nice ways that we could use that to so one, we can use it as a way to just validate that we're getting equivalent data from consumer, much less expensive equipment, and with poorer contact and things like that, right? Because again, a research system's using paste and has much better contact and stability. They're probably also putting people in a chin rest and things like that. I need to check the paper, but it's all there on ERP info. And but it would be great to extend that to the other 4 experiments, P300 and an event-related negativity. An N400, so syntax effect, and blanking on what the fourth paradigm is. And you're actually getting 7 ERPs from that, because the paradigm, I think the event-related negativity is coming out of one of the other, I think it's coming out of, say, the P300 experiment. Anyway, I don't know how much this is of interest, but it's all described here in the ERP Core open resource page, and again, they make all their raw data available. Oh, it's an N2 PC, which I'd have to look up what that experiment is. But they're, I think they're getting the lateralized readiness potential from the P300 experiment. That's the other canonical ERP. Anyway, I'd love to see us process this data using M&E. And on that earlier ERP info link, the first set of webinars that UC Davis ran was actually focused on decoding, and I've said before, I think that's really up. This is what Moab is kind of focused on, and I guess one last paper before we. So I posted this in Slack, in the research channel. So a really interesting paper from Oxford, ERP paper from Oxford, focused on decoding color, and I thought it was particularly interesting. Yeah, here it is. So it was particularly interesting because I swear somebody asked about this recently. I forget if it was in like the Neurobar after Neurogaming.
I remember I asked about it once or twice with regards to VR and identifying objects so that you can interact with them based on color.
Gotcha. And I think I said something about like, well, the way you would, the way you would do that is with, steady state and having the object flicker, basically.
Yeah.
And part of why I was saying that was that, like, I didn't think you could decode color from the ERP.
I think it might also be hard because people see color differently. Like if you talk to people with synesthesia, their idea of colors is very different. Like red is not the same across different people, and like all sorts of things get more complicated with color because it's our eyes trying to interpret stuff based on a lot of external data and make a accurate assumption of what something looks like. Whereas on or off in the terms of a flicker, it seems a lot easier for our brain to be like, something's there or it's not.
It's definitely a lot easier, right? And this is why, BCI systems all use the steady state Veep. And what's interesting in this particular paper from Mark Stokes and Kian Obry is that it's very much taking the decoding approach that Steve Luck et al. Talk about in their webinar, which was like end of I want to say it was like maybe in June. Where they were discriminating 16 directions. So it was basically like which way were moving dots going. And so there's a lot of parallels if you have a look at this paper in terms of they're picking these 16 spots on the color wheel. And as they're using LDA, right? But again, it's a nice demonstration of the same thing, but focused on color. And like I had remembered, even though I'd forgotten the context.
Also earlier tonight before the meeting, we were talking a bit about ASMR response research. Was the paper you mentioned ever brought up?
Well, so the one that what's his name? I want to say it's Hector. Just a second, I'll find it.
Because I do want to see more research with that, since like at least in social media and everything, ASMR has this one meaning that is very much not akin to the scientific like broadness of what it can do. Like I know the last time I was in a high-end EEG headset, I was watching videos, and because I know what videos caused me an ASMR response, I watched those videos and told them exactly when I was going to get the response so that they could time log it and look at the EEG and eye tracking data to see if anything changed. Because like I get a lot of visual ASMR more than I do auditory.
So the one I've dropped in, I dropped in a link to a binaural beats paper, and yeah, it's Hector Orozco Perez, along with Guillaume Dumas and Alexander Lehman, which, you know, for me is like a very, I mean, this is an awesome paper. That yeah. Anyway, it's not getting at the same, you know, exactly the same effect, but I see ASMR talked about in the same way that binaural beats. It's obviously super important to know what it is that you're actually trying to do.
Yeah.
But, you know,
I think mainly just for research and stuff, because like a lot of people seem to think that ASMR is a like form of arousal,
right.
and so because arousal is something that EG can pretty well like notice. It like at least there's been a lot of research on it recently, but
we were just talking, you know, so I mean the Bob Knight paper in Berkeley, you know, like you need to basically be you know, anesthetized or asleep to like then there's a clear marker. When you're talking about awake arousal changes, you know, it's definitely a different kind of beast. And and you know, again, like I want to be like, so just looking at like some definitions in papers, you know, several common triggers used to achieve ASMR include whispering, personal attention, crisp sounds, blah blah blah. Like so one, you know, I can the only one of those I can really speak to is the whispering, right? And that what certainly for people who are interested in studying hallucinations, as we are in psychosis, you know, research is psychosis. You know, presenting voice at close to threshold is a really common kind of paradigm. And you want to mix that up in terms of, you know, sometimes it's a real voice and sometimes it's not, right? And you're anyway, and of course you also throw in catch trials where you're not actually doing anything and you're asking, right? And there's, you know, there's some really wonderful, some great papers to read about that, especially, you know, given an auditory noise background. And, you know, basically when do people, you know, this is our goal is like, we would like to be able to induce hallucinations. Right?
And and I see a lot of places doing the audio stuff, but not nearly enough visual or other senses. Like taste is one thing we were talking about earlier tonight, before the meeting, but that like the response is not just from audio events, but audio seems to be the one that's most common for people. Even though, like, for me, if I am focusing on watching something and I notice some set of changes done in a specific way, it triggers an ASMR, and I want to know why. Like, usually it's while watching movies, and I'm like, that scene looks really cool, and the audio fits it perfectly. I want to know if I'm having just a visual one, or if it's an audio visual, or if the visual is tied to the audio in such a way that it's actually a audio ASMR response. Like, it gets really complicated, and it's something that isn't well researched, but that could definitely be applied to some of these fields if understood properly what causes them.
Sure, sure. You know, so it's something that could use the neurotech treatment, in the sense that, like, you know, catching things like you know, changes in skin conductance, and, you know, the physiological response where your hair stands up, like, is very detectable. And is pretty well understood too, right? So yeah, I'll look around for any better papers on it. This particular one with eye tracking looks cool, coupled with, you know, some EMG, like GSR like stuff. Anyway, it's getting close to 9. I wanted to, well, just in wrapping up, I know that there's, for those who might still be listening, there's a couple jobs, a couple data science jobs available. So the Mooney, the sleep device company, Muna, the sleep device company that I talked about, I know that they're looking for data scientists. You don't have to be in Paris, but that would never be a bad thing. And an Open Mind Technologies, which is a VREG company, which is based in Paris. I'm not sure if those jobs are remote, but IDUN in Zurich is hiring both a machine learning engineer and, I believe, a neuropsychologist. And that is in Zurich, which is our newest NeurotechX chapter, which I'm super excited by, just because there's, you know, certainly in translational psychiatry, there's a really famous lab there at the translational, it's like the translational neuroimaging unit, I believe, at University of Zurich. And it, yeah, we'll hopefully talk about some of their work at some point. Next week, I don't know if people, well, check the Meetup agenda. You'll see that the MIT Brains, what is it? Not yeah, Center for Brains, Minds, and Machines. I didn't want to have more B's for some reason, but it's more M's. The BMM summer course is going on for the next 3 weeks. Really interesting, and I think there's a lot of Neuromatch graduates that are moving on to this particular course. Of course, the news that Society for Neuroscience has canceled its annual meeting that's normally in October. And I see some discussion in the Twitterverse that maybe there will be another Neuromatch like event that coincides with when that meeting was supposed to take place. Since like most neuroscientists have that blocked out, that time blocked out no matter what. What else next week? The Leipzig Summer School of Neuroengineering is also going on, which I've posted that before. And that's Thomas Noshi and others that I can't recommend enough. On a somewhat personal note, it's Fedora's, it's Linux distribution Fedora's annual meeting this weekend, and we'll certainly be trying to do something NeuroFedora related. I've got a couple packages that I've been saving that basically will bring neuroimaging to the distribution in a full peer sense. Yeah. Anybody else have anything they want to drop in before we go?
I just got the fast Fourier transform stuff working on the GPU.
Nice. Okay.
I literally just got it working. No, it turns out that FFTs are really confusing, but DFTs are just a really simple sum, or like it's got a really simple series notation for it. So it works great. It's actually faster on my GPU than on my CPU.
What type of GPU is this? CUDA?
No. I'm using pixel shaders. I'm literally using like a weird. It's called GPU JS, but it just uses pixel shaders.
Oh yeah, shader based. That's smart.
GLSL.
So it turns out you can use compute shaders now on the latest versions of Chrome and Edge too. So that's kind of amazing. Because so what I figured out with this is for each channel I can just run a shader, and then that should let me do real-time multi-channel Fourier analysis.
Oh.
If I could, I found compute shader examples they could do it in like half a millisecond for like audio data. So a lot of samples. For EEG, it should be a lot easier.
Yeah, and it's amazing what Chrome and Edge and Firefox have built in now. Like if you if you go through their flags, you can actually run code in the URL. It's actually been a feature for about 10 years, but very few people know you can actually run full like JavaScript in the URL, which allows you to modify a page before you enter it. Like I put a couple.
That'll let you. Have you seen the Katamari macro? It'll modify the page so that you can roll a Katamari ball.
That's one of the ones I have saved.
That's my favorite one.
Yeah.
So
yeah.
Don't forget there's the Neuroprosthetics 2020 finally dropped their videos, and you can catch some of the talks that you might have missed, which I can't recommend enough. I'm just looking over. The Hackaday SuperCon has become the Hackaday Remotacon, and they are calling for proposals. So that's taking place in November. But if you've got projects that involve hardware creation, and yeah, it's an awesome group, and well, you know, I was very disappointed that we weren't able to do Hackaday Belgrade in May, but hopefully next year. So that's of course connected with, or you know, partially funded by SupplyFrame. Oh yeah, and I don't know if everybody saw this, but you know, BitBrain has a call for research projects with grants up to 150,000 euros of equipment for research projects in the areas of brain, human behavior, and BCI. Submit before September 21st. Sorry, I'm just catching up on what. See, any news that had come past in the last week. And there's probably more. Well, for anybody who can, there's a new group, BCI group starting in Australia. Their first meetup is August 19th. So catch that, Brain-Computer Interface Australia. And looking forward to that group. Certainly driven by researchers, University of Melbourne, that are working on some really cool things. And so I want to thank our videographers, Ryan and Alex, for all their work in getting this digitized and up on YouTube. It had to have been record time last week. Yeah, check us. You know, keep the conversation going on Slack and or Discord. And any last words? Okay, if not, see you.